Okay, great. So we'll just get started, and hopefully a few other folk will join us once we get going. So my name is Kaylee, and I'm Plantlife's community scientist in the Cairngorms, and we have with us this evening Dr. Andy Taylor of the James Hutton Institute in Aberdeen.
Hi, folks.
So tonight we're going to share with you a new citizen science project taking part across the Cairngorms. So basically it's for anyone that's really into hill walking, and it's a chance to discover more about the below ground biodiversity of mountain habitats. So we'll hear from Andy about the exciting research that's been undertaken by the James Hutton Institute to date, and what this latest study might help us uncover. But for now, I'm just going to start with a very brief introduction to the Cairngorms National Park itself, and then we'll pass over to Andy. So a few quick bits from me. So my role within Plantlife is a combination of community engagement and citizen science. So it's all about bringing people together to protect and take action for the special species and habitats that we can find in the Cairngorms. So I consider myself extremely lucky to work in such a beautiful place. And not only is it a great place for hiking, but it's also home to an amazing variety of wildflowers, plants, and of course fungi, which we'll be talking about tonight. So the Cairngorms National Park itself is the largest national park in the UK, which isn't hard to imagine if you've seen it on a map compared to the rest of Scotland. It's home to some of our rarest plant species across a varied landscape of grasslands, iconic Caledonian pine woods, and Arctic alpine habitats, which has made it an area of global importance. So as you can see here, a huge proportion of the national park sits over 600 meters. And in fact, the Cairngorms Plateau has the largest area of ground above 900 meters in the UK. So it features over 50 impressive Munros, including the famous Cairngorm itself, and also boasts Scotland's second highest Munro, Ben Macdui. So with such impressive heights, the summits of these mountains have a unique microclimate, and the harsh conditions of which plants have adapted to over time. So the greatest threat that they face is climate change. As temperatures are rising, the available area in which these plants can grow becomes increasingly restricted. So what we're doing at Plantlife is working to monitor and understand the impacts that our fragile mountaintop habitats are facing. So with more living organisms in 1 teaspoon of soil than there are people on the entire planet, it's really important for us to find out what's happening below ground as well as what's happening above. So a little bit about the project itself. It's a National Lottery Heritage funded project called the Cairngorms Rare Plants and Wild Connections project that I work on. It's running until March 2024. And really we're working to inspire local communities, landowners and visitors to the Cairngorms to take action through either hands-on conservation or citizen science like this project that we're talking about tonight. So alongside our work with meadow restoration, pinewood species reintroductions and waxcap conservation, understanding more about our mountaintops is one of our key goals. So we were really delighted when we had the opportunity to work with the James Hutton Institute. So together, we're going to be calling on mountaineers, hill walkers, throughout the Cairngorms and beyond to help us discover more about the below ground biodiversity of these iconic summits. So we have 58 Munros available for adoption from July to September, which you can climb, collecting 4 teaspoon-sized soil samples en route to the summit. You don't need to be a wild plant or fungi expert to take part. All you need is a passion for the outdoors, hill walking experience, and some confidence reading maps is also very useful. Everything else we will provide for you. So I'll talk a little bit later about how you can get involved, but for now I'm going to pass you over to Andy, who will talk more about mountain fungi, the James Hutton Institute's research, and what we hope to learn this summer. So Andy, over to you.
Hey. Thanks very much, Kelly. I'll try and share my screen now. Is that working?
Yeah, that's great.
Okay, I'll just move you out of the center of that one. Okay, evening everyone, and thanks very much, Kelly, for that introduction. So what we have been doing over the last few years is trying to increase our knowledge about below ground biodiversity in the mountains. We know lots about above ground, about the plants and the animals that are unique up there, but until we started doing the work, we knew really nothing. So over the last 4 or 5 years, we've been doing a lot more work up on various mountains, but particularly the one you see in the picture there, which is Ben An. And we've been doing various projects, but I'm going to tell you about the one that we've done on Ben An because it's the largest and the most complex study that we've done to date. So I'll give you some of the background on that, and you can see what we've been up to and some of the results that we've been getting. Here we go. So those of you who've been up in the hills might not equate fungal diversity with our mountaintops, and you maybe have come across a few fruit bodies like this little guy buried in the vegetation and not thought much of it. But all of the plants up there rely upon fungi for their survival. Most of them have fungi associated with their roots, and these are beneficial fungi, and without the fungi, the plants won't survive. And reciprocally, without the plants, the fungi won't survive. So this is it's a mutualistic association. And we've spent a lot of time looking at these because they're very sensitive to disturbances, particularly trampling or nitrogen deposition as a pollution. So you might have seen 1 or 2 of these little guys, but I can assure you, if you look long enough, you'll see lots of fruit bodies up there. And these are all collected up in the alpine zone. And some of these are quite big. So you get this crazy situation where the plants are only a few centimeters high, but you'll get the fungi about 10 or 12 centimeters high. So you get an overstory of mushrooms above the plants. So it's a very odd situation sometimes, quite amazing. But what we're more interested in fact, is what's below ground. And that's what we might call the cryptic, the hidden fungal diversity. Because most of these fungi you never see, they're below ground and they exist as fine threads in the soil, and they just go about their functions and you never know they're there. So it's only very recently with the use of molecular tools that we can extract the DNA from the soil, we can extract the fungal DNA, and we can actually identify the fungi that are directly in the soil based upon their DNA. So it's an amazing revolution that we've had over the last 20 years. And that's really what the projects have been focused on, because although we find fruit bodies occasionally, it's a tiny proportion of the fungi which are there that actually produce fruit bodies. So we're looking below ground at the vegetative parts of the fungi. And like I said, we've been looking at Ben An, and for those of you who've been up there or up any of the mountains, you'll realize that the mountains are really a mosaic of plant communities. And it's very striking sometimes that you get these very discrete, recognizable patches of different vegetation types. And I'll just show you a few of these because these are involved in the new project. So there are grasses, particularly this thing here, which is incredibly tough. This is Nardus grassland. And on Ben An, for example, there's one particular patch of this that's like football field size. It's crazy. And it's absolutely, it's wonderful stuff, although really tough to work in. So that's the grassland. Then you have the heathlands. So there's heathers and also things like crowberry or blaeberries. They are a distinct, unique component of our alpine systems, but very, very short. Those of you who've wandered through knee-length heather will be surprised that this is actually heather growing just a couple of centimeters high. So that's the other iconic habitat that we have up there. And then lastly, my favorite, I think, is the Racemitrium heath. And the Racemitrium is a moss, and you get really deep carpets of this moss. It might be, sometimes it's a foot thick, and it's just dominated by this moss. And we always think of these guys as being, mosses as being quite diminutive little characters. But up there, they are dominant in the vegetation. And this particular heath, this Racemitrium moss heath, is really, really important for some bird species. Like those of you who know the dotterel, it nests in this Racemitrium heath. So these are really important habitats for above ground diversity, but what we're interested in is what's going on below ground. So we've got this mosaic above ground, and our simple question was, is this the heterogeneity that we see above ground, how is it reflected below ground in the soil biodiversity? So we did this crazy study that we set up a few years ago where we set up 99 plots up on Ben Ann, and they were distributed across 7 vegetation types. And when we set these plots up, there was 4 of us, did over 24,000 meters of ascent in 2 weeks in July 2018. And I actually wanted to do a bit more, so it was actually 3 times Everest. But I think we've done enough. So we set up these 99 plots, and we have done a lot of work up here. These are the sort of mini plots that we established in each of the vegetation types. And we looked at all sorts of things above and below ground. We looked at the plant communities, composition, the biomass of the plants, and then we also set up some assays that we could see what was actually going on. So we have nutrient probes that are collecting data on the nutrients. We dug 36 soil pits, so we've got all sorts of parameters for the soils. And then we have these things at the top there called eye buttons, which recorded the temperature in the soil over a year. And these record I think it's every 15 minutes. So you have the temperature of the soil at all these different plots. And then for the functional aspect, we did this great thing. It's a global tea bag assay. And it's you bury tea bags, and it's got to be these 2 tea bags from Lipton's. And they must have made a fortune, because there's been hundreds and hundreds of experiments using these tea bags. And basically you bury the tea bags in the soil at 5 centimeters deep, and then you leave them for a given amount of time, and then you go and dig them back up again. So you weigh them before you put them in, and you weigh them afterwards, and you can see how much has been lost during the period that they've been in the soil. And it's a measure of the decomposition in these systems. And also we use these lollipop sticks as also a long-term measure of decomposition. These were buried for a year in the plots. And so if you think about it, that's 594 tea bags we buried that we had to go and collect after 4 months. And we collected all but 3. 3 disappeared. And for the lollipop sticks, we collected 98. 1 had been chewed to bits by a hare, we think. So it wasn't bad. It wasn't a bad recovery. So you get all this data that we can then look at all sorts of things relative to what's going on below ground. For the biology directly, we used a technique. It's a bit like going to the supermarket and scanning the barcodes on your shopping. And it's exactly the same thing. We extract the DNA, and we use a portion of the DNA as a barcode to identify all the organisms that we're interested in. And in this case, that was bacteria, fungi, and then what's called eukaryotes, and that's everything with a nucleus. So similar to us, for example. So it gives us a really good image of what's actually in the soil. Okay, so that was the protocol, I can say, for that project. And I really just want to give you a snapshot of where we're at, because we're still processing the data. And for the fungi, I'll give you some figures here. From all the plots, 99 plots, we ended up with over 5.5 million barcodes. So that gives you some idea of the numbers that we can actually generate from these new techniques. And what that translates to in terms of species, and this is just for the fungi, so it's just under 3,500 species of fungi that we have recovered from these plots. So an immense number. If you think about the fruit bodies that we've seen, you'd be really, really lucky if you get 50 or 60. Okay, so what we found is just under 3,500 species of fungi up there. For the bacteria and the other guys, that's a different story. There's many, many more. Okay, but I'm just showing you the fungi because that's the focus of the new project. Just to give you some idea of how the numbers are distributed between the vegetation types, the one on the left here, the SNO, that's the late snow beds, and they are incredibly rich, both with respect to the fungi and all the other groups they've looked at. So you've got this sort of gradient across this diagram here down to Kaluna Heath and the subalpine heath. Okay? So they do have very, very strong differences between the vegetation types. And the communities differ massively between the vegetation types. And that's really what the new project is all about. Okay? All of this data comes from one hillside, an amazing hillside, all the same, but it's only from one hillside. Okay, so just going back to the original question, how is it reflected? They're incredibly intimately linked. You could take a sample up there, and if I looked at the biology underneath in the sample, I could tell you what vegetation it's under. It's so linked, closely linked together. So the basic story from this is that the soil organisms are the biggest component. It's a massive component of the biodiversity in our mountains. And we expected a lot, but we didn't expect as many as we got. Okay, so it's a huge component of the biodiversity. And what you can imagine is, as Kaylee said, in a teaspoon of soil, you've got all these numbers of organisms, but it's not just the numbers, it's the species. There are hundreds of species underneath your feet, and you don't even know they're there. Okay, so that's the Ben Ann study. The new one, which I think is absolutely fantastic, is so that was like I said, that was just one Munro. So what we want to do is to get soil samples with your help, and thank you so much for being interested in taking part, is to get soil samples from all 58 Munros in the Cairngorms National Park. And we'd like the samples from each of the main vegetation types, so from under the grass, from under the shrubs, and under the moss. And on the instruction sheet that you'll get, we very clearly describe these. There's photographs to help you identify what you're looking for, and we've given you very good coordinates where you should find these vegetation types. So we've also included what you might call a potluck sample. So if you've been up on the mountains and you think, well, I wonder what's in the soil here, here's your chance to find out. Okay, so we'd like 3 samples from designated vegetation types, and then there's one for you to choose where it comes from. And what can we expect, really? Well, certainly based upon what we've got so far is we will vastly increase our knowledge about what's up there, because like I said, before the Ben Ann study, we knew almost nothing. So if you multiply that up 58 times, of course, the coverage is less than what we've got at Ben Ann, but will hugely increase what we've got. And I was saying to Kaylee that we should run a sweepstake on the numbers of fungi that we should actually expect and have a prize for that, which would be quite fun to do. Okay, so we'll massively increase our knowledge about this biodiversity, and we're incredibly lucky here because these systems are some of the most pristine systems we have left in the UK. And this is one reason why there's such a very close link above and below ground. Any other system you go into, it's had hundreds, if not thousands of years of management. Okay, so there's all sorts of effects have gone on which have changed above and below links. Here, they're still pristine, and it gives us a fantastic tool. If besides all the knowledge, it gives us an amazing tool to say how things should be. Okay, because in Scotland, we're in a brilliant position where things are still very, very good. In the Lake District and in Wales, things are not like this. We've been down there and got samples from there, but that's another story. Okay, so we can say what's been lost and how we might go about rebuilding these things. And that's really, really important. Just a wee note about what's next. When we have got all the 58 done, over the next few years, we may run the project to all 282 Munros in Scotland, which should just be a unique undertaking. And that would just be absolutely extraordinary. Okay, so that's me. Thank you very much. And thank you very much for making this possible.
Thanks so much, Andy. I'm just gonna chat through a wee bit about how people can get involved if anyone's interested in signing up that hasn't already done so. I'm recognizing a few names of folks that have already signed up, which is brilliant. So great to have you here. So let me just share my screen with you again. 2 minutes. Okay, right. Hopefully, did that work? Is that up for you, Andy?
Yep.
Yep. Perfect. I'm seeing thumbs up. Excellent. Thank you. So yeah, so if you do want to get involved, this is just a wee bit about you know, the joining in process. So basically, it's really simple to adopt your chosen Munro or Munros for the summer. We know that quite a few people have gone for, you know, one or two just because they're on a kind of obvious walking route. So it makes sense to go for both of them if you're up on the summit anyway, and you've got the next Munro just a short walk away. So we have all 58 listed on our volunteer management system, which is called Better Impact. So that can be accessed through a website link, which I'll pop in the chat feed here for us. And it's also on the end slide of this presentation, so you've got it there. And you'll see all 58 listed, so you'll know which are still available if you're looking to bag one. So anything marked with a 1 is still available, and anything marked with a 0 has already been adopted. So you'll be able to see it's updated live, so you'll be able to see straight away which ones are still there and sign up for those. So if you see one of your liking, you'll just need to fill in a little registration form. It's very quick and simple, and that's just at the top of that web page there for you. So it's all online, very quick and easy. And once those details are provided, I can send you out your survey pack. So we'll have all your details for that. So what does the survey pack consist of? So it's quite a small A4 envelope that we send out to you. So after you've signed up, I'll get that all packaged up and sent in the mail. And it contains everything you need for doing your sampling. So instructions, you know, it's got the little kits. I've got there the sample pots, and they're really quite small. So it's, you know, not going to be a huge task having those in your hiking bag. And as well, as Andy said, we've got little sort of identification guides to help you when you're out. So yeah, everything's nice and small. It's not too complicated, and yeah, really easy to hike with. So you don't need to worry too much about size of those. We'll also give you an aerial and an OS map of your sample points, just so you can see clearly those 3 red markers where we've got the different habitat types to sample from. And then of course you get to choose your 4th lucky dip within that area too. So a few quick things to just bear in mind before heading out on any surveys and before you head out for your hike. We'll be analysing soil samples, so it's just to remember you're not collecting any above ground material such as plants. So definitely just the soil is fantastic. There's a few photos we'll need from you, so we'll take photos of your actual sample spots, but also we would just love to see how you get on with the adventure. So if you've got any snaps from your hike, or just, you know, if there's a group of you sampling, we would love to see it. So please do keep in touch and share those with us as well. And it's important to return your samples in the post just as soon as possible. So once you've done your hike, pop those in the post as soon as you can. But if you can't get them straight away, just pop those in the fridge for a wee bit, but definitely do not freeze, and just let us know when they've gone in the post, just so that we can kind of keep track of everyone's progress. And yeah, lastly, and most importantly, if you do join us for the study this summer, we hope you have an amazing time and enjoy your hike in the beautiful Cairngorms. So I think that's us wrapped up and just open for any questions. see if we've got anything in the chat
Yeah.
feed. People are also welcome to put on their cameras and, and microphones and things if they'd like to chat. But I'll just see what's on here. Okay, so you mentioned that 3 of the sample areas were for different types of vegetation, but how do you select those areas? On the top, is it EO based?
Can you stop sharing, please, Kaylee, and I'll show how we've selected them.
Here we go.
Yeah. Okay. So, does that come up?
Yeah, that's great.
Okay, super. Okay, so for each Munro, we have both a satellite image, and it's got the superimposed vegetation, the National Vegetation NVC map on it. And so Andrea, who's jointly running this, has selected the areas of the correct vegetation type within the NVC maps. So you've got both the OS map showing where the plots are, but also the satellite image. Okay, and in the instructions it said if that's not a good point, then choose an area close by that is more suitable. So just in case it's on a cliff face or something like that, please don't put yourself in any kind of danger at all. Okay, so that's how we've chosen them. So these are pre-chosen, and this is the approach that we've taken at Ben Ann. And I think out of the 99, I think we only had to move 2 any kind of distance. So, you know, just maybe 5 or 10 meters. So it's not the position per se which is important, it's the vegetation. And in the guide, we have a good description and good photographs of what you're really looking for. And one of the things that we'd like you to do is to take a photograph of the plot that you actually sample, because then we can verify it. And that's particularly important with the potluck one, because we're really reliant upon you telling us what was there. Okay.
Great, thanks, Andy. So we've got a couple other questions. So the next one is, can the results of this research be applied to practical habitat management in any way? So, you know, in the Cairngorms, for example, or elsewhere.
You want me to answer that?
Yeah. Well, I guess, you know, what it does tie into the information we're hoping to find out, because at the moment we know very very little.
Yeah, we know extraordinarily little, I have to say. And one of the things which is becoming more apparent is the role of the soil biodiversity in maintaining our systems. And if we can acquire this baseline data, which is absolutely missing, it really has a very practical purpose, as well as being, you know, just from the knowledge point of view, massive. Because we actually applied for a project to do something similar on a pollution gradient from the north of Scotland down through central, the southwest, the Lake District, and down into Wales, to look at what has been lost. Because if you go down to the Lake District, it's grassland. You know, they have lost an extraordinary amount of their alpine zone, effectively. And we had a meeting a couple of years ago at the Institute in Aberdeen, and we were talking about climate change and the effects of climate change. And one of the wardens from the Lake District said, "We don't have any impact of climate change." And we were like, "Huh?" And he said, "Yeah, we've got sheep." So sheep are the dominant force in the Lake District, and they have just grasslands left. And the climate change that we're seeing elsewhere with the migration of plants and the change in plant communities, which is happening, they don't see, because they have lost the components of those communities. So it has a very practical application.
Mm-hmm, definitely. And I guess for us on the Rare Plants and Wild Connections project, really between now and 2024, really it's an information gathering exercise for us. We really just want to know what we don't know at present, so that we can make that action going forward and see what can be done. So yeah, hopefully, you know, judging by the data you had in the previous study, I think you're going to have plenty plenty to work
for sure.
with.
Well, one of the things we realized when we were doing this and thinking about the differences in the heights of the Munros is really important. Over the, I'll need to dig this up from my memory, but over the 282, I think it's 400 and something meters of difference between the lowest and the highest. And what that allows us to do, if we get information from them, we can actually look at the impact of temperature change effectively, because that 400 meters represents something like 3 degrees change. So you can effectively, you can model what would happen with a 3 degree shift in space. So it would be an amazing data set to be able to do that.
One of the other questions we've got is about the weather. Will the weather conditions matter? So if it's lashing down with rain or it's a sunny day, will it still be fine?
The only way it matters is it won't affect what you get out, but it does affect processing, because the wet samples are more difficult to deal with. But that's not something that you should worry about. That's for us to worry about. But it's just a bit of an aside. Yeah.
Great. And we've had another question pop up. Do the underground mycelial networks in the mountains connect with plant roots in the way they do with the trees, tree roots? So I think this is linking on to the Hidden Life of Trees book, which is on my to-read list, but I've heard very good things.
Yeah, there's a few books like that coming out now. Yes, is a simple answer. All of the plants, well, all of the plants except mosses, it's amazing. All plants, all terrestrial plants, there's always exceptions, never mind. The great majority of land plants form what are called mycorrhizal associations. And the mycorrhizal comes from myco, fungus, rhiza, root. And basically it's fungi invading or colonizing the roots of plants. And these are mutualistic associations, okay? They both benefit. And in general, they are absolutely obligate associations. Neither can survive without the other. And they are particularly important in harsh environments. And it doesn't get much harsher than up on the tops there. So all of the plants have fungi associated with their roots. And some of these fungi can colonize many different plants, different species, even different orders. And so they link lots of things together. And we know that these mycelial networks, as they're called, are incredibly important. And it's one of the things that a lot of plant ecologists don't like, because it messes up all the competition arguments about competition between plants, because you've got fungi in the soil sharing everything. So yeah, it's an interesting question.
Brilliant. I'm just checking I've not missed anything. As I said, if folk want to pop up on screen, if anybody wants to ask us anything, that's no problem. Just checking through. Julie, who asked that question, says that's amazing.
It is amazing, I think. Woodlands are, it's actually what I work on. I usually work on mycorrhizal fungi, particularly those of our trees, so pines and birches and things.
I thought I'd just come in because it just feels a bit unsociable sitting with our cameras off.
Yes,
I just talked to myself and Keely.
Yes, please.
So I just wanted to ask your comment when you said there's a few books like that around. Yeah. I mean, I found that was an absolute eye-opener, that secret Hidden Life of Trees book. I mean, I know it anthropomorphizes trees a bit, but it was it did make me think about trees in a very different way. But you said there's a few books that have been written that way. What did you mean when you said that?
Well, there's another book by Merlin Sheldrake, and he does it from the fungal point of view.
Oh, I've seen this one.
Yeah.
It's a black book, isn't it, with a colourful cover on.
With a name like that, you couldn't expect anything less. But it's a gift of a name. I haven't read it, I have to say. It's meant to be good. Factually, it's meant to be good. Yeah, which I think is very important.
So somebody had read that book, said to me recently that even seaweed are connected with fungi, and that seaweed colonize fungi. Fungi colonize seaweed, if that's the right expression, I don't know, to stop it drying out when it's in the intertidal zone. And without fungi, it would die because it would be dried out. Which was another amazing fact about fungi.
I haven't heard that one.
Oh, maybe.
I have a question for Kaylee, because I might have missed it. By when do we need to have the samples mailed in by?
So we've said the very end of September. So 30th of September, you really need to have done your Munro, have it in the post latest. I'll be keeping an eye on everyone signed up, and if I'm not seeing things in the post before the last week in September, I'll definitely be giving people a little nudge and a reminder. I guess because we've got it across 3 months, it's the sort of thing, I don't know, you might sign up and think, oh, I'll do that next month, and you know, before you know it, you're a week before the end of it. So I'll definitely be sending little reminders every now and again if people have got their sample packs but haven't hit the hills yet. They'll get a little nudge, a gentle nudge. And then Andy, I guess, you know, do you want to talk a bit about that process of analysis at the end, how long that's going to take you? So I think spring next year, we're hoping to kind of revisit things and see where we're at.
Yeah, for sure. I mean, it takes a while. There's what is it, 232 samples in total. So they have to be processed, and that takes a wee while. And then once we've extracted the DNA, that has to be processed and sequenced, and that can take 5 to 6 weeks. Just that part. Yeah. Going to just go back to the fungi and the plants. Sure. The Racemytrium moss that I showed you, we've done quite a lot of work on that, because it's one of these iconic habitats that has declined massively in the UK. We've done several studies on it, and we looked at what was happening, or tried to work out what was happening. And it's very heavily impacted by increasing nitrogen. So with nitrogen deposition from the atmosphere, once the level of nitrogen deposition gets above around 10 kilograms per hectare per year, the decomposition of the moss just goes up like this. And what's actually happening is, although the nitrogen increases makes the moss grow faster, the decomposition from the bottom goes even faster, and it catches up. So those amazing mats that you saw, they fragment, and other species can come in. So you've got something like, you know, on top of Benan, it can be, I don't know, 15 centimeters deep. When we were down on Snowdon and the surrounding mountains, I was struggling to find patches, you know, that were that across and about that thick. And they used to have exactly what we had. So it's been incredibly heavily impacted. And it's one of the possibilities why the dotterel has declined so much, because the habitat's just gone.
And is the reason that Racemytrium decomposes so quickly, is that because the nitrogen feeds the bacteria that then decompose the Racemytrium? Is that how it works?
Very good.
Oh, it's
actually the fungi.
Oh, the fungi, sorry.
Yeah. Bacteria is really weird. When you think about decomposition, everybody thinks about bacteria, but they're hopeless.
I should know.
No, that's fair to say. I'm a mycologist, but the fungi they play the biggest part in plant decomposition because it's the chemistry of the plants is really complex, and the lignin, which gives plants their strength, I should say. It's one of the most complex natural substances there is. And bacteria have very limited capabilities for breaking down lignin. Mosses don't have lignin, but they have analogues that are similar. Do you know about the story of coal and why we've got coal? No? You know about this, Julie?
Only what I learned in school.
Yeah, that's what I was thinking.
When they're trees and they all got compacted.
Yeah. But why? Why didn't they decompose?
Were there no fungi?
There were lots of fungi. Fungi have been around for 1 billion years. They've been here long before trees. Yeah.
There's no nitrogen.
No, fungi didn't have the necessary enzymes in order to break down the lignin, so they couldn't access all that amazing wood that was lying around. Okay, and there was a study. There's always been the idea that once the fungi had evolved the enzymes to break down lignin, that was the stop of the Carboniferous period when all the coal was laid down. And there was a study about 5 years ago, or a bit longer now, and they managed to date when the fungi evolved the enzymes to break down lignin, and it was right at the end of the Carboniferous. It was brilliant. So it was an idea that had been around for ages, and then they actually managed to show that the evolution of the enzymes was really at that period. It was great.
That's amazing.
Yeah.
So we got any other questions from anyone? We've got a few folks sitting quietly. So you're welcome to say hi if you're there. But yeah, any other questions for myself or Andy?
Have you got many people signed up, Kaylee? How are you doing for your 50 Munros? Have you got
Yes.
Are they all?
So we
all
have.
taken?
I think when I checked, we have 20 of the 58 signed up, which is a great start. So we've still got plenty up for grabs. But yeah, it's been really great. Lots of walking groups and things that have started getting involved, which is brilliant to see. So yeah, it's a good start. Of course, because it runs till September, we might just get a few coming in a little bit later, just because it's running for a few months. But hopefully, start of the season, it would be great to get them all assigned and get everyone's packs out, so they've got all 3 months to plan their hike.
Oh, and when will we get our packs so I can plan my walk?
Yes. So I'm planning on sending the first bunch. So those that have signed up already, I'm planning to send them either tomorrow or Friday. So basically, towards the end of this week. So they should come in the post pretty soon. So yeah, they're just sitting waiting to go out. So yeah, anyone that's signed up already, they'll get there soon. And then beyond that, it'll just be as and when folk come in. I'll just package them up and send them just once they get assigned. So it shouldn't be too long a wait, basically. Great. Okay. Well, if we've
Sorry, can you pop that link, the BetterHelp link, in the chat, just in case somebody isn't signed up who's here?
Yes, I had that ready to go. Thank you for reminding me.
No problem.
So we've got that there, and yes, it's really easy to sign up through that. And if anyone has any trouble, they can get in touch with myself via email or anything like that, and I can help them with sign-ups. It's a bit of a funny spelling, so I will also put that in the chat so that people have that there. But it's just in case anyone has any trouble, or they've not received a pack, because it will be myself sending everything off and checking all our sign-ups. So that's also my email on there if anyone does want to get in touch about that sign-up process. Great. Okay. Well, I think if there's no more questions, I think that's us for tonight. But yes, hopefully do feel free to share with any other like-minded walkers, hikers, mountaineers, anyone that loves a good Munro. It would be great to see you take part. So please do feel free to share what you've heard tonight, and we'll also, because we've got a recording of this, have that available soon for you. So anyone that's registered tonight, there's a couple of people that couldn't make it, so I'll just send around an email to everyone with that link for those that want to catch up. Great. Okay. Well, thanks so much, everyone. Hopefully we'll see you on the mountainside at some point.